Okay, so final session of the day. I just wanted to welcome our distinguished panelists, and this session is going to be, dealing with the future of transplantation and where we think this field is going. So what I thought we would do is I would give a couple of minutes for each panelist to introduce themselves and also to bring up an issue that they feel or a question that they feel is important for the future of transplantation. So maybe we'll start from the left.
Hi, I'm Ali Zaranpar. I'm a transplant hepatobiliary surgeon at the University of Florida. I think the biggest question for us is, and I think this was brought up during my talk a little bit earlier today, is how do we prove that sort of a personalization or individualization of treatment is actually going to be better for patients without having to prove it in every individual? How does an entity like the FDA, how is that going to look on any changes that we make in this sort of precision, personalized type medicine?
Thank you.
Hi, I'm Karen Sherwood. I'm director of the HLA lab here with James Lan, and I also run the Genome Canada Transplant Consortium, and have a research aspect as well. So, yeah, I mean, one of my biggest questions in the last few years, big science has become a big thing, and it's sort of like the antithesis of precision medicine. You have big science just to answer a single patient's question, and I think we need that approach. But how we integrate that knowledge into the clinic is going to be its own beast. I mean, the FDA has regulatory control over that. But how do we get to the point where we can say to the FDA or the Health Canada, so we've done all this research and we're going to integrate and we've designed this panel of omics. And this is how it's going to change clinical management. That's like the first step before we even get to the regulatory stages of understanding what we need to do for our patients.
Well, I think we've, in 2 days here, realised that we've still got gaps and there are unmet needs for patients. And we are not doing a great job at moving the field forward. And we're talking about personalised medicine. So that means that if you've got a treatment for person A, you've got a different treatment for person B. But at the moment, we've only got one treatment. So we've got 3 different phenotypes of patients and we give them TAC, mycophenolate and PRED. This one's different, so we'll give them TAC, mycophenolate and PRED. And this one's different to the other 2 and we'll give them TAC, mycophenolate and PRED. So I think what I'm outlining here is that we're not going to go forward unless we bring in new agents into the clinic. And we're not going to bring new agents into the clinic unless we start doing different types of clinical trials. And we're not going to be able to action those clinical trials unless we cooperate as a field globally and do those trials in a cooperative way. So I think that's a change of mindset at a lot of levels, I think, for a lot of people. But I think that's the way forward. And I think at least people are talking about it. I think with the next steps, why don't we get in and do something? Yeah.
Hi, my name is Anne Halpin. My background is that I'm a laboratory technologist for many years. I won't say how many. And but I recently finished a PhD with Laurie West, and so I've been thinking a lot about ABO blood groups, but also I think about all of these things in the context of the practical, because I'm a bench technologist by background. So I think I'm maybe thinking a little smaller picture than some of the things that have come up already, and we've only really touched on ABO a little bit in this past 2 days, but we know that there's inequity around ABO and allocation and access to transplantation. And we talked a bit about, some ways to overcome ABO barriers by removing ABO. But I think, we know that also ABO incompatible transplantation is something we can do. But the methods we use really lack precision. And so thinking from a very practical standpoint, how can we as a community try to introduce more precision and more harmonization of the methods that we use around this to enable more of this incorporated into the trials that we're doing, because it's really not something we do a lot in Canada. I know other parts of the world are doing this better than we are, and some parts of Canada are doing this better than we are. But how can we, as a community, advocate for this? Where does it fit into the laboratory? Who's going to do this testing? And how can we propel this forward to better serve our patients and allow for more precision with better matching with larger donor pools?
This has been great. Chris Blosser, transplant nephrologist at the University of Washington. A couple of things come to my mind as I hear other experts talking now and over the last couple of days, and that includes that there are advances happening outside of immunosuppression, including cellular engineering, including xenografting. And it leads me back to a larger question of how can we maintain or even claim greater trust with society that is required to do all of this work. And that includes working directly with our patients and hearing what they're seeing and what they're experiencing. The science has moved forward so quickly, while the patients are continuing to be one. And each one of those experiences are valuable. And I do think that in order for us to advance the field, we're going to need to involve our patients in the implementation science that is so necessary in the next steps.
Yeah, I completely agree with everything that's been said so far. I'm Dave Harriman. I'm a kidney transplant surgeon here in Vancouver, and I think the xenotransplant was a good one that I was thinking about as well. But I'll go with precision organs. With CRISPR being a technology, and with everything out there, there is a chance for once organs are outside the body to do modifications, whether that's human organs and changing different things, or whether that's a xeno organ. Hopefully one day we look back and we say, oh, it was barbaric that we were taking organs out of people and putting them into other folks. Hopefully we have solutions that there's a dialysis chip that you can implant into your arm, or there's some other things that we're not doing these things anymore. But this is the best we have at this point in time for kidney transplant specifically. But I hope we get beyond that sometime.
So all excellent points. I saw the three speakers on the panelists on the left were talking about personalized medicine and regulatory FDA. And actually, James and I had a discussion the other day about FDA approval and, let's say, Health Canada approval. We're discussing about, is there a way, because it sounds like each country is on its own. Some countries may be able to approve things. Companies are probably having to spend a lot of money for each individual approval. Is there anything that could be done to streamline that? Because we have issues right now with approval for drugs and so on. I can see that when we go into personalized medicine, that's going to be an even different level of potential struggle. So any ideas from the panelists about what needs to change to actually accommodate that?
I don't know anyone at the FDA, so I can't presume to speak for them. But I think the FDA is going to have a lot of trouble with AI. I think they're already having a lot of trouble with AI and implementation and approval of things like that in the clinical practice. But really, what they need to try to figure out is, how do you approve a process, right? How do you can't just approve, for example, a drug that is individualized, but maybe you can approve the process of making an individualized drug and allow that company to make an individualized drug for every patient using that specific way. There needs to be kind of a shift in thinking in that respect.
Yeah, I think I'm turned on that way. Yeah. So yeah, that's a tricky question, and it's really one for the bar afterwards, I think, when we can really work that out. But I think there's a few things. So there is harmonisation. So I know I'm from Australia, so we have a thing called the Therapeutic Goods Administration, which is a poor man's version of the FDA. But the FDA and the EMEA and the TGA, and I'm assuming the Canadian one, have a policy of harmonisation. So basically what one does, if you get over the big bar of the FDA, you're pretty much going to make the other bar. I think, though, the regulators must be scratching their head at a broader issue. So if you think about transplantation, I don't think the regulators go to bed at night worrying about how they're going to solve that problem. They probably doesn't get on their radar really. But where it has been designated an orphan disease, and they're now looking, and there's increasingly people are looking at how to treat rare diseases. Now, rare diseases are rare, but if you add all the rare diseases up, they're reasonably common. So a lot of people have a lot of morbidity or death from a rare disease. It's different to someone else's rare disease. And this is an area where I think medicine's making big advances now, where a lot of this data is happening. And the FDA are now having to work out, well, how do we make an approval process for something that's rare, where it's easy for hypertension and, you know, and things like that. So that's one challenge. But on the other hand, we've got to do our bit too. So everyone works in their own environment. So the thing about then is that not only is once you've licensed it, you've got to get reimbursement, whatever system, whether you're in a national healthcare system like Canada or Australia or in the US, where you've got different types of payers. And they're going to require data to show that it's beneficial. And there's got to be a cost effectiveness to that, because these treatments are expensive and there is a limited budget. That's a reality. So I think on our end, we've got to start thinking in novel ways. How can we provide evidence that shows that this is actually helping, that we're getting better life years, better quality of life, better return to work, those types of things we can do. And I think there's better ways of collecting that data now than used to exist, and we can do that. And the FDA will take real world data in certain difficult situations.
Well, I'm blissfully ignorant of the FDA, so I don't know that that's going to change. Yeah, but I actually was thinking, when everyone's answering much smarter than me, answering this question so far, about Chris's point about patient, about what I heard from you is patient engagement, and I don't know if the FDA has what a voice that brings to that or not, but I think that we've not done that well historically in transplantation, and almost every single time a patient says, is asked a question, what risk would you want to assume, or what do you think this or that? They almost never say the thing I would have predicted them to say. And so I think that maybe as part of this whole conversation, also patients need to be much more involved in communicating what they would want from the FDA. And perhaps, I know obviously they're not going to sway the FDA completely one way or the other, but I think that involving patients in these conversations might actually work in everyone's favor, including the researchers.
Maybe to follow that, I think that there is high value in having dialogue with patients, recognizing that every patient is a single person experiencing that journey, and bringing in more than one patient and having a diversity of perspectives enables a much greater perspective overall. The NIH now requires that every application for some type of funding include patient proposal or patient perspective. And I think that's a part of what we need to consider in all of our writing, including our manuscripts. I would suggest that we start to think about, how do we write for patients' perspective, including a lay language summary and or involving them in the writing of a paper. Secondly, when it comes to the research we do, one thing that I did when forming the research center that I helped to lead is to create a community engagement committee made up by multiple patients that provide feedback on the strategy that we're taking for our research, as well as advocating about our work into the community, so that it's bi-directional. And I learn something every time I talk with them.
The only thing I'll add is United States and Canada, there's a little bit different standards in collecting outcomes data within transplant centers. In the US, it's very organized, and all data on patient outcomes is reported to a central database. And in Canada, we don't have that. So it's up to individual transplant centers, I believe, to actually collect their own data and to have data that's interpretable that we can actually put into research. And I think here in Canada, we can do a better job of making sure we have that data available, and it's in a way that we can readily harness it, so that we could actually say that this is how our patients are doing with these interventions. It's an important thing to have a really good database with each transplant program.
So that's an excellent discussion. And as you were saying, that's quite interesting with the data. Should there be almost like a development of best practices? Should journals require the data to be collected in certain way, and that certain type of data be included? Should there be almost like a standard operating procedure of how certain types of research in transplantation be conducted, so that again we can all understand this in a easy way, and that patients and studies from different centers can be almost translatable, I guess.
I think that would be a fantastic idea from our perspective here, because if we have organizations saying that you have to collect with certain standards from in Canada here, that would get government to get behind that and throw some money at that, so we could hire people to be able to do these jobs of collecting the data, which it's a huge job. And if it's the clinicians who have to do it, it just doesn't get done very well, because there's other things they have to do. If it's trainees that have to do it, it just won't be done very well. It really needs to be a dedicated team, and I think we've lacked funding for that in the past. And if we have the funding and people say, "Hey, you have to do it this way," I think that'd be fantastic.
And that might also help with this almost never-ending issue of getting ethics and then sharing data across geographical borders. I always find that hilarious, because if I was 1 mile on the BC border, I couldn't share my data with my research collaborator who was 1 mile on the other side of the border for no other reason other than someone drew a line at that point. I was like, no, your patient's different from mine, and I don't want to share my data with you. So yeah, ethics is always another issue, and we've been working with a number of international groups where we've been able to standardize the way that each group in that collaboration has collected the data, but now we can't analyze it, because unless we analyze it within center, we can't analyze it as one large data set because of ethical reasons. So if someone else took the responsibility of that, say the journals or the funding agency said, okay, this is upfront, you have to be able to, one, standardize your data collection, and two, have a strategy in place that allows, you have a templated ethics, you have a templated data sharing agreement that all institutions sign off on at the beginning of your funding, then you don't spend 4 or 5 years trying to get that in place. And ultimately, in every experience I've had so far, failing.
You just have to be careful about requiring things, because in the US, the data collection is required but not funded. So have to come up with the money somehow.
But could case be made that the investment put into standardizing data collection and putting infrastructure in place to collect it would then help gather better information, which would translate into better development of treatments and all the testing and all the other stuff.
I'm going to quote James here. One of his favorite saying is, "If a problem can be solved with money, it's not a real problem."
So me being a laboratory person, I'm going to ask a selfish question. To get to the future of transplantation and personalized medicine, what do the panelists think, say, the HLA labs need to do better? How do you see the role of HLA labs in this process?
Okay, I'm the oldest HLA person in the room probably, so I'll start with that. I think HLA, any clinical lab, but HLA labs, obviously in this example, we sit on a gold mine of information that we have no time to do anything with. We have a biorepository. HLA labs are a biorepository. I have a freezers full of spleen. You're asking, where do you get spleen? I don't know how many thousands of spleens. Like, we have thousands and thousands of sera samples. We have so much that we could do, but we don't have the resources and we don't have the time. And moving forward, we're probably not even going to have hardly technologists in our lab. We have this looming shortage of technologists that we need to train and retain and do all these things. So I think we have to advocate better for ourselves to get more resources. And I think the clinical lab has always been in the background. We're not visible. I wasn't on your slide earlier. You have to add lab to your slide. Nobody thinks about the lab, so we need to advocate. We need to labvocate for ourselves, and we need to be more visible. And I think it's not a natural thing for the clinical lab to do, but I think if we don't do it, we won't get what we need. And I think we have a lot to offer because we have samples and information. And I think we could also collaborate better with our national registries, like in Canada, CTR, and get more information. We have our, some information in there now, but not as much as we could, post-transplant in particular. And so I think there's a lot that could be done. But yeah, resources are going to be a problem for sure, and people.
The other thing I was thinking was generally hospital labs just do the routine work, and that's great. As a lab person, you do good lab work, but I think we need to try to change the way that hospitals see these labs and allow for a bit of more flexibility in terms of clinical research, right? We're not trying to turn them into basic science labs, but a little bit of flexibility in terms of how the funding flows, how the resources are used, space is used, because it's going to be these kinds of specialist lab places that, in collaboration with the physicians and collaboration with the patients and other research groups that really help drive the clinical research forward. Like the clinicians, like Dave and Chris and Ali here, they're interested, but also limited in time. So you can't have it land on their shoulders, all of it land on their shoulders to drive research forward. So the lab also has to play a responsibility in taking some of that on.
I definitely agree with Karen's point that having a clinical research arm to a lab is highly valuable and often informs the way the clinical part of the lab works and vice versa. And I guess related to that concept is harnessing the strengths of your bioinformatics team or team members. I think that the presentations today are a great example of how we haven't done well, and we often get trapped in our own perspectives and then can't see beyond that, and being able to really power with and partner with those people that have the capacity and often do much more efficient work with the data and then the samples. So there's opportunities that surely are available for that.
The only thing I'd add to this is what I've found very valuable is actually even walking into the HLA lab as a surgeon. I've really welcomed the relationships we've forged, and I've welcomed the crosstalk. If there's something wrong, that having this teamwork and having these connections and being collegial and showing respect to every single team member is just such an important thing that we all need to do. Historically, we haven't been that good at that. Communication's really lacking in healthcare a lot of times, and that's pretty scary and sad to think. But I think that's one thing, is just the HLA lab's not a silo. It's an active team member, and all those members are team members of the entire broader transplant team.
Okay, perfect. Excellent discussion. I just wondered if there were questions from the audience for the panelists. Please go ahead.
Hi, thank you. And I would like to ask another question to the panel, and that's related to what Anne said yesterday regarding live kidney donors, and that there's not a lot of studies being done on the follow-up of live kidney donations. So I was wondering if any one of you is engaged in any activities related to studying live kidney donors, or what would you think would be needed to do more research or do more follow-ups from your perspective?
Yeah, that's a really good question. So I think we've always got to remember that, and I always tell the people on our staff that the live kidney donor is having an operation they don't need, that everyone else in that hospital is in the hospital to have some procedure or have some treatment that they need, but the donor is there for someone else. So I think, therefore, we've got high obligations to ensure that what we're doing is at least no harm to those people. And if we don't have long-term follow-up, then I think we put ourselves at risk of letting these people down and letting ourselves down. So I know It's difficult, I know, in Australia. So what we did, we set up a donor registry. So we've got a thing called, we've got ANZ Data, which is the patient registry, and we've got ANZ Otter, which is the donor registry. And so the catch is that following those people up has been difficult. So, but at least we've got every patient down on a registry. So at least if you want to go back and follow them up, and there's other ways you can do that, you've got a national registry of the donors, which is at least a starting point. At our own hospital, we were working this out. So first you said they had to come back every year for a follow-up. So they'd come back, you'd see them post-op, you'd say everything's fine, they'd come back at 12 months, and then they're back at work and they're sitting in your waiting room for 2 hours. You finally see them for 5 minutes and their blood pressure's normal, they're looking fit. You might tell them you might need to lose a bit of weight, but you're fine. And they don't come back ever again because they've just wasted a whole day of work. So what we then did was we put them back to their primary care doctor and asked, and we'd write to them, the primary care doctor, to say, can we have the blood results? And we just said what we wanted. And then at 5 years, we send a letter out to the people who at 10 years. So at least, and we've followed them, and we get about, interestingly, we get about 50 to 55% response rate. At 5 years, people will come back with data. Sometimes it's funny, you get a 40-year-old guy and you get a handwritten letter from their mother saying, I read his letter and I sent back the result. That's quite amusing, but it's only 55%. But then I've looked that's pretty standard. So that's something you've got to know about the follow-up, that half of the people aren't going to do what you say. So I think that behoves us to do the best we can to make sure we've got an ability to follow these people.
I'll jump in next. I think as one of those people, I think it has to be easy, as you said. Like it can't be something you have to take a whole entire day off of work, and so now we know we can do virtual medicine. Like we do this all the time, and so COVID taught us some good things, and I think that's one of them. So I do think that's a good idea. I agree with you, and I think also maybe. And I don't know, I think there's, at least in Canada, there's a lot of variance from program to program. What is the post-donation follow-up? And I think maybe that's true in Australia. I assume it's true in the US. And so I think choosing a gold standard, what are the tests we need? What could we follow so that the data are at least comparable? As Karen say, you have a dog's breakfast of data. You can't do anything with it. But if you are collecting the same things, and I think if it's explained to donors up front too, like this is really important to you because of this. And I think it's hard to strike the balance of not scaring away donors to feel like, oh, these are all the risks to you, and therefore we need to monitor you. Although they do kind of scare you a little bit with all the information. But I think that it's I think in Canada we have, at least for KPD, we could be for sure using KPD for this purpose, and we don't. And that bothers me. I've been emailing people saying, do you want to follow me? Who wants to follow me? And I think it's a problem that we don't have a facility to do that. And so I think there are some really low-hanging fruit here. And I think it not only would be good for donors, but I think for patients, as was said yesterday by the recipient video that we saw, she really worried about her donor. And I think from speaking to the small number of patients that I get to speak to, some of them are actually afraid. They don't want to ask donors to come forward because they're so afraid that they're going to harm their donor. And I think if we had better data, we'd actually have more donors, not just because of donors being comfortable, but because of recipients being comfortable.
So I'll just finish off on that. We do quite a bit of living donor kidney transplants here in Vancouver, and we are actively engaged in donor research. We have some post-op pain studies for donors. We have some longitudinal GFR studies that we're looking at. There's some retrospective analyses to see those donors over time who have gone on to dialysis and subsequent transplantation. What were the factors common to those folks that actually made them get on dialysis? I mean, one of the problems with doing donor studies, though, in general, is that this is such a healthy population. They're sort of boring to study in a lot of ways because they, as a group, they tend to do really, really well. And if you actually look at the donor literature, it's really boring to read because it's not that there's nothing really new coming out in it. So it's a bit of a problem because I totally agree that we need to keep making sure we're ensuring the health of all these folks who are giving a piece of themselves they otherwise would not need to do. It's really important to preserve the health of donors so that they have confidence in the whole system.
First of all, I will give two perspectives of my research. Like I am using a fake blood that has bovine hemoglobin, and I'm perfusing human organ. I'm just perfusing. I'm not treating any patient. It's an isolated organ. We'll perfuse certain amount of time, wash them out. Hopefully, there will be no bovine hemoglobin inside the human organ before transplantation. Number two, I'm using carbon monoxide. Of course, when people say carbon monoxide is really, really scary, but we know that we produce carbon monoxide every single second in our system. Without carbon monoxide, we won't survive. Now, my question is how, if this knowledgeable panel knows how FDA, Health Canada works, basically, do they have all this expertise in place in order to make a decision? And how academic expert in the respective field facilitated their decision-making process? What's the mechanism actually? Who is seeing, who is FDA, who is Health Canada? Particularly at this stage, I have no clue. And it is becoming questions in coming future that how quickly we can facilitate the clinical trials processes.
I've actually been involved in organising a meeting with the FDA to try and improve how we can get new drugs into the system there and talk about new forms of clinical trials. So my experience of that, and so if I start with the FDA, I think those people they're very well trained. They are experienced at what they do. They've got certain sections, so they're not, you're not doing transplantation one day and antibiotics another. So they sort of group things together that are similar. The other thing you've got to realise that everyone swears about those idiots up there making all these regulations, but everyone works in the environment to which they've been placed. And their issue is that safety is number one, sometimes irrationally so, and that they have to respond to legislation. So there's not much room for common sense here, or let's be serious here. Everything's done on a legislative framework, and that's the same for the TGA. And when people have explained that to me, then you understand sometimes why they make those decisions. There's not much leeway to how they make other decisions. So you've got to get a pretty high level change to make them consider different types of data, because that leaves them exposed in a way to, if something goes wrong, they're up before a Senate committee or something. They've got consequences for them. So I think the first thing you got to do if you want to deal with them is you've got to work out, well, what's the environment they're in? What triggers them, and what things have you got to do to get them to make a decision that you think is going to help the field? That takes a long time, but you need to make those thoughts, or you're not going to get anywhere.
Any other comments from the panel? No. I guess my question would be about that is when clinical trials are being designed, or anything that has to then go through an FDA approval, is there some kind of a consultation up front that occurs to? Okay.
Yeah, there is. So the companies traditionally will put it to the FDA, and then the FDA will go and think about it, and they'll say, I want to do this. And so there was a period, how crazy things in. So they used to, when they did the I think it was the belatacept studies, they looked at what was the most common immunosuppressive protocol in the United States, and it was tacrolimus, mycophenolate, and prednisone. But the standard of care was cyclosporin, mycophenolate, and prednisone. So they insisted that the comparator would be cyclosporin. And the field thought that was a bit crazy. And it took some years after that they then changed what was standard of care. And you could respond to this better than I can, but I know some of those things are infuriating. But they do have consultations, and they'll say you need to do this, or this study won't cut the mustard, or you need to change your endpoint. What's your primary endpoint? And when you're looking at that, so some of those things they're very prescriptive about what the company needs to provide. Yeah.
You said it well, Phil. The only addition I would make is that tacrolimus has never been FDA approved for transplant, for kidney transplant, and that's why cyclosporin was required as the comparator arm, because tacrolimus has not been FDA approved, and so it had to be FDA approved in order to merit that comparator arm. And that's very frustrating because, like you said, tacrolimus is the standard of care, and yet it never gained that either upfront or afterwards. So the ongoing conversations are iterative.
Very interesting, very illuminating. I think we're out of time, so we'll end the session here. I really wanted to thank the panelists for excellent discussion and thoughts about the future of transplantation. Thank the audience for questions.